Hi everyone. In this video, we're going to look at some of the ways in which signaling pathways inside of cells can be amplified and controlled. If you haven't had a chance to watch it yet, I recommend pausing and checking out the video on signaling pathways and receptors first before you go on with this one. We're going to start with a look at how multi-step pathways provide a means for signal amplification. Let's consider a GPCR, which binds to a single ligand activating the associated G protein. But instead of a single G protein, let's see what happens if 3 G proteins are activated. Each activates an adenylyl cyclase, and let's assume that each of those produces not 1, but 3 molecules of cyclic AMP, resulting in 9 molecules of pKa being activated from a single ligand. Now, these numbers are actually much smaller than those used in cells. One ligand binding to one receptor can actually activate as many as 10 G proteins, each of which can activate an adenylyl cyclase. Each of those enzymes can produce many molecules of cyclic AMP, resulting in a response potentially millions of times greater than the original stimulus. Now, proteins in signaling pathways are frequently regulated by phosphorylation. In this example, we're going to look at a set of proteins that function downstream of a receptor tyrosine kinase. And in the example here, you can see that the ligand is already bound and the receptor has been activated. If you haven't watched it yet, I recommend watching the video on receptors to see the details of how that works. The protein that was activated by the receptor is one called a protein kinase. In simple terms, a kinase is an enzyme that phosphorylates something, just as we saw with the receptor tyrosine kinases, which phosphorylate tyrosine residues on themselves. This is the first step in a kinase cascade, in which each step is a kinase, which is activated by phosphorylation, and goes on to activate the next by phosphorylating it, until we reach the response. You can think of adding the phosphate as switching the enzyme on, and so removing it, which is carried out by a set of enzymes called protein phosphatases, can switch the kinases back off, and so turn off the response. As well as turning components on and off, multi-step pathways provide a lot of additional opportunities for regulation and control. Assume we have a ligand which activates a receptor, and the downstream relay molecules activate a specific response that we'll call response 1. Since different cells produce different proteins, we can have a situation where the same ligand can activate different receptors in different cells, leading to different responses. Now, if our blue relay molecule here activates others, we can have a situation where pathway branching leads to other responses, and we can even have multiple pathways with multiple ligands communicating with each other, either amplifying or inhibiting each other to produce different outcomes. Varying gene expression, and therefore protein expression, as we'll see in a later section, is critical for allowing cells within a multicellular organism to specialize. And what we have here is really just the tip of the iceberg in terms of how just a few signaling components can lead to a variety of cellular responses. Thanks for watching, and if you haven't had a chance to check it out yet, please make sure to watch the video on signaling pathways and receptors.